Welcome to Physiology Fundamentals with Dr. H, where we make physiology fun by understanding the fundamentals. Today's fundamental is the process of osmosis, making water move, like parting the sea.
That's not correct, Dad. It's osmosis.
Oh wow, what's osmosis?
The diffusion of water.
Thank you. Well, I'll have to look into that. You mean like this?
Yes.
Okay, well, my students always tell me spelling isn't that important anyways, but we'll check it out and we'll dive into it. Today's topic is the process of osmosis, making water move. So we're going to discuss the topics of diffusion, osmolarity, tonicity, and osmosis. First of all, let's understand the process of diffusion. In diffusion, we need to have a difference in the concentration of something. So for example, in our diagram here, we have sodium in water. Yeah. On one side of the container, there's a lot of sodium. On the other side, there's basically no sodium. So we have a difference in concentration for sodium. So what will happen? If we allow enough time, and if sodium is able to move from one side to the other, eventually sodium will move. Why is it doing this? Well, it turns out that each of these sodium molecules is moving around randomly. So it could take a step this way or a step that way. And each has an equal likelihood. But if there are more molecules on this side than this side, then only molecules moving this direction will affect what's going on over here. So the only choice, if it goes this way, it doesn't affect the concentration over here, but going this way, it does. So some of the molecules will randomly decide to move this direction. Yeah? And the number of molecules moving that direction is going to be more than the number of molecules moving this direction if there are more molecules over here than there are over here. So what is the result? The result is, if we allow enough time, randomly more molecules are going to move this way than are moving this way, and eventually, if there's enough time, the concentrations of what is dissolved in the water will become equal. This process of going from high concentration to low concentration is called diffusion. Now, another concept to understand in terms of concentrations is the osmolarity. Okay? Osmolarity is the total concentration of all dissolved particles, especially if we start to pay attention to things like sodium chloride. Yes. If we discuss the concentration of sodium chloride, this only counts as one molecule. Yes. But it turns out when we put sodium chloride in water, it will separate into a sodium ion and a chloride ion. Yes. So we need a different way of measuring the total number of molecules when we're talking about things like salts, like sodium chloride, which will separate into two ions or individual particles in the solution, right? So we use osmolarity especially for this, right? So because in osmolarity, we want to know the total concentration of all the dissolved particles, okay? And this includes the ions, okay? So, for example, if we had a sodium chloride solution, right? If we put in a solution, and we know about chemistry with concentrations, you can find descriptions of this elsewhere. Let's say we put in a 150 millimolar solution of sodium chloride, okay, that would be the concentration. That's the number of sodium chloride molecules that were added. What would the osmolarity be? Well, it would be roughly 2 times that, because each one of these is going to give us 2 particles. Yes, so the osmolarity would be 300 milliosmoles per liter to equal a 150 millimolar sodium chloride solution. And this is meaningful when we start talking about what is in our plasma. Tonicity also has to do with osmolarity. We're talking about the number of particles, including the ions, that are present in a solution, but there is one key difference. With tonicity, we need to have 2 solutions that are separated by a membrane that is permeable to water, yes, but not permeable to something in the solution. Okay, so when we look at this, let's look at this example here. So on this side, okay, we have water and we have a permeable molecule. All right, so both water and whatever this permeable molecule is are capable of diffusing to the other side down their concentration gradients. On the other side, we have water and we have something that cannot diffuse. In this case, it's sodium. So sodium is not able to diffuse to the other side down its concentration gradient. So if we allow enough time, what sort of things will happen? Well, one thing that will happen is this permeable molecule will diffuse down its concentration gradient until it is equal. Yes. So when we have something that's permeable, if we allow enough time, it will become equal on both sides of whatever this membrane is. On the other hand, if we have something that is not permeable, if we allow a lot of time, the concentration is not going to become equal on either side of the membrane based on diffusion. Okay, so this is an important concept. So when we are talking about tonicity, okay, we are only interested in the osmolarity or concentration of particles that cannot cross the barrier. Yes? So the only molecule that is important here in terms of tonicity in this example would be the sodium, okay? Because it does not diffuse. Okay, so it will maintain its concentration on one side of the barrier. So, if we flip this around a little bit, it's kind of an interesting concept to think about. We focused on the concentration of the sodium, but actually water can have a concentration too. So let's look at that. Okay. When we have water on this side versus on this side. Okay. By the way, when we are comparing these 2 solutions, again, tonicity has to do with 2 different solutions. Okay. The side that has fewer of the impermeable molecules, the side that has fewer of the impermeable molecules is called a hypotonic solution. Yes? In this example, this side has fewer of the sodium molecules. The side that has more of the impermeable molecules is called hypertonic. Yes? In this case, we have more of the sodium molecules. So we can have a hypotonic solution that has fewer impermeable molecules or a hypertonic solution that has more impermeable molecules. So that's where the name comes from, hypo and hypertonic. But we can also think about it the other way around in terms of the actual concentration of the water. So if we look at it on this side, the hypotonic side, there are not a bunch of other particles that are taking up space. Yes. So there is actually more water on the hypotonic side. If we look at the hypertonic side over here, okay, what we see is, since we have all these other particles in here, there's less room for water. So we actually have less water on the hypertonic side. Okay. So when we talk about tonicity, two types of solutions. Yes, we have hypotonic. Yes, what does that mean? Okay, it means there is less of the impermeable solution, which means then there is actually more water in terms of concentration. Okay. On the other hand, if we talk about hypertonic solutions, what does that mean? That means that there is more of the impermeable particle and therefore less water. Yeah. Okay. Now, if we have two solutions that have the same concentration of the impermeable molecule, then we call these isotonic solutions. So when it comes to tonicity, we need to understand hypotonic, hypertonic, and isotonic solutions. Why is this so important? Well, if we think about it, if water has a concentration, the water concentration is higher on this side, which is the hypotonic side, okay? And the water concentration is lower on this side, which is the hypertonic side. If this membrane is permeable to water, and it is, and it's not permeable to this, yes? So these molecules of sodium, in this case, are not going to diffuse across, but water can, right? Water can and will. Why is water going to diffuse across? Because it's going from its high concentration towards its low concentration, okay? So actually the diffusion of water from a higher concentration to a lower concentration has a certain name, and we call this osmosis. So what is osmosis? Diffusion of water from a higher concentration solution, which happens to be a hypotonic solution, versus to a lower concentration solution, which happens to be a hypertonic solution. So yes, the tonicity of two solutions separated by a membrane drives osmosis between them. Okay. As long as there is a difference in tonicity, one side is hypotonic compared to the other side being hypertonic, water is going to continue to try to move down its concentration gradient from hypo to hypertonic solution, and that is osmosis, and it is driven by tonicity, which we keep in mind is the osmolarity, the total number of particles, but only those particles that do not diffuse across the cell membrane. Because if they did diffuse across the cell membrane, we would just wind up ultimately with isotonic solutions, and we would not drive osmosis. Okay, so this process will continue until something opposes it, or until we reach the point where the two solutions are now isotonic. So where in the body is osmosis important? Well, here are three important examples that we will talk about. The first are in your blood vessels. Osmosis is an important process for keeping the balance of fluids between what's in your blood vessels and what's outside your blood vessels. Cell membranes. Osmosis is very important for keeping a balance of the fluid inside your cells versus outside of the cells. And along the tubules in your kidney, where osmosis is very important for controlling the concentration of your urine so that we can keep the concentration of our plasma at the proper levels. So in a small blood vessel called a capillary, osmosis turns out to be important. So a real quick review over blood flow in our body. Of course, the source for driving the blood around in us is the heart, and the heart adds blood pressure, and we call this pressure hydrostatic pressure to the blood, and that's what drives the blood through the cardiovascular system, through our blood vessels. Now, when we reach these really small blood vessels called capillaries, where the job is to exchange things like pushing oxygen out and maybe pulling some CO2 back in as waste products, the force that pushes the fluid out is that blood pressure. It's called hydrostatic pressure. So in a capillary, when we reach the capillary, there's a force pushing some fluid out into our tissues, and that's called hydrostatic pressure. It doesn't happen in earlier blood vessels because they're not very leaky, they're not very permeable. Capillaries are permeable to pretty small things like sodium and glucose and water. And so we push all these things out, and of course oxygen goes as well. Now, one thing that capillaries are not permeable are to proteins that are in the blood, like the key protein albumin. So we have albumin in our blood, floating around. We get to a capillary, it's still in the blood, floating around. Now in the capillary, we push some fluid out, so we actually take the volume down of the blood that's in the capillary, but the albumin particles stay put. So if we take the same number of particles and put them in a smaller container, if you would, in our capillary, okay, we have increased the concentration of albumin. Okay. Now, albumin is not permeable across this membrane here, which is the capillary wall. So that creates an osmotic pressure, right, to drive osmosis. Like we said, we have an impermeable molecule on one side, and we don't have it on the other side, so that is going to draw water back in to the capillary. And this is really important because if we kept pushing fluid out of our capillaries and didn't have a way of pulling it back, we would have fluid accumulating outside of our blood vessels, and this is called edema. So osmosis is super important in your capillaries to make sure we don't have edema. We need to pull back the fluid that we are pushing out in our capillaries. Another place where osmosis is super important in our body is across the cell membrane. Okay, so here we have a cell, and inside the cell are some impermeable molecules. All right, so the orange dots here represent impermeable molecules, and they're both inside the cell and outside the cell. And they have equal concentrations inside and outside in this illustration. So what that means is they are not going to contribute to osmosis. These would be isotonic solutions, comparing the extracellular solution to the intracellular solution. Since we don't have a difference in the concentration of this impermeable molecule, we don't have a difference in the concentration of water, so water isn't going to diffuse. This is the situation we want our cells to be in. We don't want water going into and out of our cells. So we want our cells to be in an isotonic extracellular solution, meaning it has the same tonicity as what's inside of the cell, right? That's important. And generally, that's what our cells live in. But what happens if we have our cells not in that situation? Well, what if we put our cells in a hypertonic solution where we have more impermeable molecules outside in terms of concentration than we do inside? Well, that of course in turn means that there's a higher concentration of water inside than outside, and so water is going to diffuse by osmosis out of the cell, and that cell is going to shrink up, and we don't want that to happen. So a hypertonic solution causes osmosis to drive water out of the cell, and the cell shrinks. Now, if we look at the opposite situation, if we have our cell in a hypotonic extracellular solution, where we have fewer impermeant molecules, and therefore we could say a higher concentration of water, water is going to diffuse into the cell by osmosis and cause it to swell up, and it may even explode. So osmolarity and osmosis are very important to our cells. We generally want to live in an isotonic solution so that the cells are not shrinking or swelling. That can be a real problem for things like blood cells and brain cells if they start to shrink and swell. The last of the situations we're going to talk about where osmosis is super important in the body is in the kidney tubules. So the kidneys have many jobs, but one of the jobs is to control the osmolarity of urine. Yes, so we need to control the concentration of things like salt and waste in the urine. Now, how are we able to do that? Well, in our journey to convert plasma to pee, so please check out that video to help you understand this, we're going to go along this tube, and we call this tube the nephron. We don't need to worry about that right now. But one of the goals of the nephron is to be able to change the osmolarity of the urine it produces. Okay? So how we do this is in one part of the nephron, we are able to pump out sodium. Okay? And some other salts, but we'll focus on the sodium. So we have a lot of sodium outside of the tubes. We don't have any permeability to water, so water does not follow. So now what happens is we have created an osmotic pressure, right? So when we come around this tube, okay, and we are coming down this part, which is called the collecting duct. That's not critical. So we now have a choice. If we make this permeable to water, then osmosis will drive the water out here into this hypertonic solution, because what is inside of the tubule is now a hypotonic solution, because the sodium is not permeable across this membrane, and water is under some conditions, right? So osmosis is very important in our kidneys to allow us to be able to reabsorb a bunch of water. Now, if we don't want to reabsorb a bunch of water, then we just make this membrane not permeable to water, and we won't do that. So very important for controlling the concentration of our pee. So in summary, diffusion is the random walk of particles from a high concentration to a low concentration. Osmolarity is the total concentration of dissolved particles in a solution, and this includes the ions like sodium and chloride. Tonicity is similar to osmolarity, but is the concentration of only the impermeable particles in the two different solutions, right? So if we have a difference in the concentration of these impermeable particles, then we have different types of solutions. And these types of solutions are hypotonic. In a hypotonic solution, there is a lower concentration of the impermeable particles. In a hypertonic solution, there is a higher concentration of impermeable particles. And if we think about the water concentration, it is just the opposite. So what tends to happen is water is going to diffuse from hypotonic solutions to hypertonic solutions. And finally, we have isotonic solutions. If we have these equal concentrations of all impermeable particles across the membrane, we will not have any osmosis. Osmosis, then, is the diffusion of water from a hypotonic solution, which has actually more water or a higher water concentration, to a hypertonic solution, which has a lower water concentration. Osmosis is important in the body across the small blood vessels, the capillaries, cell membranes, and kidney tubules. I hope you found this helpful. I'll see you on the next one. Thanks for watching Physiology Fundamentals with Dr. H. Please like and subscribe, and don't forget to get outside and go for a run.